Hello, everyone. Well, what a hard act to follow, these three talks. Thanks for being here. I try to stay on time so we can answer questions at the end. My name is Sepideh, and I'll be talking about connectome dynamics today. And I want to start by thanking a lot of amazing collaborators for sharing generously their data with us. So I'll start with the view that we're very familiar with, which is the view that fMRI gives us on connectome dynamics. We know that these slow dynamics that we can observe with fMRI explain variability of performance within participants. So here to the right is one example of the brain of one participant. Sorry, I'm trying to get the mouse here to show. That is a much more modular state prior to successfully detecting a threshold level stimulus compared to when the participant missed that stimulus. We also know that variability across individuals is explained in terms of performance, and that connectome dynamics are brain traits and that they're genetically determined to a quite large degree. But fMRI misses connectome dynamics at the most behaviorally relevant time scales, and what we know much less about is about dynamics and electrophysiological data modalities. And so I'll focus on that today. Recently, we analyzed about 1,000 participants from the Minnesota Twin and Family Study research. We looked at their resting state data and canonical frequency bands, source-localized the data to the Desikan-Kiliani atlas, and leakage corrected them. Then we apply modeling to get at recurrent states. You see here the alpha band as an example over about 24 seconds worth of data, and the colors represent the most likely states at the given recording time. And so from that, we can derive the overall occupancy of every state, so the fractional occupancy, and then the probability of every transition between all possible brain states, and so that's the transition probabilities. And so both of these features, fractional occupancy and transition probabilities, are multidimensional as they are derived from multiple states, and so we can represent every individual participant as one data point in this state space. In this participant space. And then the origin is defined by surrogate data that lack veridic dynamics. And so every individual participant's position relative to that null can also be investigated for associations with cognitive abilities. And just to remind us here that we're looking at a traditional twin study. These are my twins, and I think the suits basically just sum up their relationship to each other pretty well. So when we look at fractional occupancy and transition probability in monozygotic twins, we see much closer similarity in that multidimensional space. So this is just one pair of the subjects to give you a sense of the similarity of their fractional occupancies as compared to an unrelated pair of participants. And we saw the same relationship, this genetic impact being true in all frequency bands except for the delta band. When we apply variance component modeling to get at the amount of heritability, about 40 to 60%, depending on frequency band, of these dynamic temporal features are heritable. And just for comparison, we applied very similar statistics earlier in fMRI data from the HCP data set, and the heritability there was in the low end here, around 40%. Also paralleling what we knew from fMRI, but this time for rapid dynamics, is that even though we analyzed a very exhaustive set of spatial features, we found no evidence for heritability of the spatial arrangements of the states. So, for example, how the strength of connectivity is distributed over the edges and how that changes from state to state, as well as how the modularity is different between the different states, is not heritable. And so then we, in a follow-up study, we took the data to a multivariate correlation analysis between latent factors of a cognitive battery and then principal components of fractional occupancy or transition probability in the different EEG frequency bands. And so we found a significant mode of association that on the cognitive side is carried by this factor broadly representing a variety of executive functions and nonverbal reasoning. And as interesting is that what we did not find in this association, for example, crystallized abilities are not associated, memory is not, at least in this young adult sample. Reward processing, for example, is not. And then on the EEG side, the association with the minor exception here, fractional occupancy, is almost exclusively carried by transition probabilities. So the way the states are sequenced is what's associated with cognitive abilities. And again, the spatial features, so the spatial arrangement, the actual instantiation of the states in individuals that was not found to be heritable is also not associated with cognitive abilities. So I think the really exciting view emerges when we put the slow dynamics and the fast dynamics together, because it really gives us a different view of the connectome. And we have used the word connectome multiplex to an analogy to multiplexing and communication systems to really clarify this view. So let me start with the motivation here. I think Rita Hari really beautifully sketched it out that even for simple sensory perception, we're dealing with a world that is inherently multi-time scale. So here she's showing that the temporal frequency with which the different parts of the tree are moving, and so the movements that we're receiving through our eyes is a function of the mass of the respective areas. So the very nimble leaves are vibrating quite rapidly, and then the heavier and heavier parts move more slowly. Similarly, if you look, for example, here at the air pressure waves of someone singing a piece of a nursery rhyme, you see that the amplitude modulation of the phonemes are quite rapid, and then the amplitude modulations of the syllables and the prosodic stress point are slower and slower, and all that is embedded in slower time scales at the sentence level and at the paragraph level. And so inherently, cognition from perception to language really requires the brain to accommodate multiple time scales at every moment in time. And each one of these processes requires communication between distributed brain regions. So definitely the connectivity architecture of the brain ideally would be accommodating this. And we do find evidence for that. So I'm starting off with very rare data that combines fMRI with concurrent recordings of intracranial EEG in humans. These are similar patients as we heard in the talks prior. And this was graciously shared with us from Louis Lemieux's group at UCL. So here's for one participant an illustration of what we did and what we found. And so for the electrodes available, we generated a band-specific instantaneous connectome and did that for every time point here. And you can do this with phase coupling or amplitude coupling. It doesn't matter, we find the same observations. From the concurrent recordings, we also have, just lost the mouse here, I think. Don't know where it went, but okay. So on the other axis, you see fMRI time and the connectivity time windows. And so what you see, the points on the plane that you see are denoting pairs of time points within which connectivity patterns were spatially similar between EEG and fMRI, more so than expected by chance. And this is corrected for all of the multiple comparisons in this plane. Now, if EEG connectomes and fMRI connectomes would represent in this dynamic manner the same underlying neural processes, then we would expect higher density of spatially similar states along the diagonal, which is the physically corresponding time. I completely lost this mouse. The mouse is not letting me click. Something happened to the mouse. All right, thank you. So we would expect higher density here along the diagonal, or if fMRI was lagging, we would be somewhere close to the diagonal, but evidently that is not the case. And so even if we HRF convolve the underlying EEG time series, we still find no closer temporal synchrony between the spatially similar states. We replicated this in healthy participants, scalp, source-localized EEG, fMRI. And again, what I'm showing here is the ratio between the spatially similar states, so occurring in EEG and fMRI. Off the diagonal means in asynchronous time versus synchronous times. And you see that we have fairly strong evidence for a lack of effect here. So we're really at a ratio of 1. And so to conclude, similar connectome states do occur, but they don't occur synchronously, and they really seem to represent different neural processes. If you heard that amazing talk this morning by Alessandro Gozzi, that would probably not surprise you, but here we go. All right, just to show you what this really looks like in the brain, I'm showing this time series in the scalp data. Okay, so this is scalp EEG-fMRI. I'm showing you correlations of the ongoing spontaneous fluctuations in the fMRI and EEG patterns to this map. So in this case, we're looking at just one single network, the visual network. And so this is simply spatial correlations in terms of similarity of the fMRI signal to this map. And during the times where the correlation is really strong, expectedly, as you would hope, the fMRI is showing a connectome state that's dominated by the visual network here to the top left. And we do see spatially similar patterns in the different EEG frequency band, a bit noisier, but you can kind of make out the visual network here. But what's interesting is the time points of particular high correlation with the visual pattern does not seem to be bearing any temporal relationship between the different time scales of connectivity. So if you do the same for another network here, the default mode network, again, as you would hope, the time points with particularly high similarity to the default mode network are indeed representing a connectome state that's dominated by the DMN here to the bottom right. And again, you see spatially similar states like that in the EEG. Again, at different time points than in the fMRI. But what's really astonishing is when you put that side by side is that at one physical moment in time in this concurrent recording, connectivity at the infraslow time scale, which is best measured with fMRI, is dominated by the DMN pattern here, whereas the connectivity in the EEG delta band at the same physical moment in time is actually dominated by a different state that is high with the network connectivity of the visual system. So without going into any methodological detail, and that's really my last data slide, we did something similar, not just with individual networks, but all possible combinations of neurocognitive networks as templates, and then these are color-coded maps, essentially, or color-coded time series that show which of those states best matched the brain recording at that moment in time. So this is just recording over about 2.5 minutes from one participant, and the different colors simply represent different brain states, okay? So in fMRI, down here, you see that the brain switches one time from one state to another, but during the same period of recording, you see a lot more state switches in the EEG frequency bands. And in fact, the speed of those state switches is scaled with the speed of the underlying oscillatory frequency. And again, you don't seem to see any obvious relationship between the states in these different time scales. So just to clarify that in a cartoon, what that means is if we imagine all of the possible states of the brain as some sort of state space here, and the infraslow connectivity trajectory that's best mapped at fMRI might be moving in one corner of this space, we have multiple other trajectories simultaneously that are much more rapidly evolving. And so what that means, again, is that at one physical moment in time, we will not have just one dynamic connectivity pattern, but we will have essentially a stack of connectivity patterns concurrently. So just to summarize, and I hope we have, yes, some minutes for questions. Functional connectome dynamics can be measured with non-invasive EEG as well in source space. To a reasonable degree, it's noisy, but it gives us amazing information, including individual variability and cognitive abilities, because we're really tapping into the important time scales now. And I think we really should take that leap and use these kind of methods, MEG and EEG, more often, because it gives us a new view, I think, to think about connectome dynamics not as one stream, but actually as multiple parallel streams that are ongoing at the same time. And these streams do have some degree of spatial similarity in terms of their states, and I didn't show any data for that, but we also observed that the statistical properties of sequencing, so the transition probabilities, for example, are very similar between fMRI and the concurrent EEG. However, the streams unfold at different speeds, and they are really appear to be independent of each other. And so I think that that is how the brain perhaps might be responsive to the multi-time scale nature of the environment that we're dealing with. And with that, thank you.